Imagine a single bacterium released from a leaf or a sea spray droplet, carried by wind currents across continents before it settles in your bedroom. Today we trace that journey, uncover how these microbes survive the sky, and reveal their hidden influence on clouds and ecosystems. Dust storms lift soil particles that carry bacteria from the ground into the sky. These airborne microbes disperse over long distances, settling on distant surfaces and water bodies. Sea spray aerosolization releases viral particles and bacteria when waves break against the shore. The resulting droplets evaporate, leaving microbial cores that remain airborne for hours. Leaf shedding during autumn releases fungal spores into the air as part of natural litter cycles. These spores can travel with wind currents, colonizing new habitats when they settle. Microbial entry into the atmosphere is a key step in global nutrient cycling. Atmospheric transport allows microbes to influence cloud formation and precipitation patterns. Understanding these mechanisms helps predict ecological impacts of climate change on microbial dispersal. Future research will map specific taxa and quantify their roles in atmospheric processes. Pause 0.8. Airborne microbes travel through the atmosphere, guided by physical forces. Turbulence creates chaotic eddies that lift and disperse particles. Convection rises warm air, carrying microbes upward into higher layers. Wind shear can redirect these rising plumes horizontally across landscapes. The boundary layer near the ground limits vertical movement until turbulence breaks it. Computational fluid dynamics simulations reveal how airflow around buildings shapes plume paths. These models show that building geometry can trap or accelerate microbial transport. Vertical mixing diagrams illustrate how microbes spread from ground level to the free troposphere. Distance traveled depends on wind speed, turbulence intensity, and particle size. Understanding these mechanisms helps predict exposure risks in urban environments. The scale of transport ranges from centimeters in indoor drafts to kilometers across continents. By quantifying airflow patterns, we can model how far microbes travel under different weather conditions. Pause 0.8. Airborne microbes face intense ultraviolet light that can damage DNA and proteins. Many species counter this by producing pigmented molecules like carotenoids that absorb harmful wavelengths. These pigments act as a sunscreen, dissipating energy as harmless heat before it reaches cells. Desiccation threatens microbes by removing water that destabilizes membranes and denatures enzymes. Microbes mitigate drying by accumulating trehalose, a disaccharide that stabilizes proteins and lipids. Trehalose preserves membrane integrity during extreme dehydration, allowing cells to survive air transit. Oxidative stress arises from reactive oxygen species generated in the atmosphere. Antioxidant enzymes like superoxide dismutase convert radicals into less harmful molecules. Spores add protection with multilayered coats that block oxidants and shield DNA. Melanin-like pigments in the outer coat further scavenge free radicals, enhancing survival. These combined strategies illustrate how microbes adapt their biochemistry to survive long airborne journeys. Pause 0.8. High-volume air samplers collect large volumes of ambient air in a single run. The sampler draws air through a filter that traps microbial cells on its surface. After sampling, the filter is transferred to a sterile container for downstream analysis. Quantitative PCR amplifies target DNA sequences, allowing precise counts of specific taxa. This method uses fluorescent probes that emit light proportional to copy number. Metagenomic sequencing captures all DNA present, revealing community composition without bias. Sequencing reads are assembled into contigs and matched to reference databases for taxonomy. Bioinformatics pipelines then generate abundance tables that can be plotted as heat maps or bar charts. Flow cytometry can rapidly count individual cells, providing a complementary estimate of biomass. Combining these techniques gives both quantitative and qualitative insight into airborne microbes. Pause 0.8. Microbes travel through the air as tiny, resilient particles. They carry enzymes that break down organic matter in soil and water. This decomposition releases nitrogen compounds into the atmosphere. Nitrogen gases then diffuse upward, linking terrestrial and marine ecosystems. Some bacteria possess surface proteins that act as ice nucleators. When these proteins encounter water droplets, they trigger ice crystal formation. Ice crystals grow into cloud particles, influencing precipitation patterns worldwide. Thus, microbes help regulate the planet's hydrological cycle through cloud chemistry. Their movement across continents creates a global network of microbial exchange. This connectivity supports nutrient redistribution and genetic diversity in distant habitats. Atmospheric transport also disperses spores that colonize new soils. These spores initiate fresh cycles of organic matter turnover elsewhere. Pause 0.8. Microbes travel through the air we breathe in every day. When inhaled, they follow the respiratory tract to the lungs. This route allows microbes to interact with immune cells lining the airway. Such interactions can help seed a diverse microbial community inside us. A balanced indoor environment often contains fewer microbes than outdoor air. Comparing indoor and outdoor loads shows how building design influences exposure. Higher outdoor levels introduce new species that may expand the host microbiome. However, some airborne microbes carry proteins that trigger allergic reactions. Allergen thresholds are measured in parts per million and vary by individual sensitivity. Understanding these limits helps design ventilation systems to reduce risk. Balancing microbial exposure with safety can promote respiratory health over time. Pause 0.8. Microbes enter the air through dust storms, sea spray, and leaf shedding. They are transported by turbulence, convection, and wind, surviving harsh conditions via protective pigments, spore coats, and trehalose accumulation. Advanced sampling and sequencing reveal their distribution, while their ICA nucleating activity links them to cloud formation and nutrient cycling.